Hello, I'm Alice Gerlach, and here's the latest update from Spectrum Autism News. Loss of autism-linked gene dampens social interactions in animals, by Holly Barker. Mice and zebrafish that lack the autism-linked gene GIGI-F1 show atypical social behaviors according to a new study. The findings tie variations in the gene to social traits in autistic people, the researchers say. Among the genes strongly linked to autism, GIGI-F1 ranks as the second most commonly mutated one in people with autism and related neurodevelopmental conditions. But little is known about the gene's contribution to autism traits, says Bo Jiang, professor of forensic medicine at Huazhong University of Science and Technology in China. Initial findings point to a role in regulating social behavior. Deleting the gene blunts social memory in mice through mechanisms thought to be mediated by impaired insulin signaling. GIGI-F1 is also implicated in other pathways, including autophagy and mRNA regulation, that are often altered in people with neurodevelopmental conditions, says Jeremy Veenstra van der Wiel, professor of developmental psychiatry at Columbia University, who was not involved in this study. But it had not previously been rigorously studied for its impact on neurodevelopment and downstream behavior, he says. Zhang and his colleagues combed sequencing data from SPARK, a project that aims to collect genetic and clinical information from 50,000 families that have at least one child with autism. SPARK is funded by the Simons Foundation, Spectrum's parent organization. They identified 7 new GIGI-F1 mutations and pooled the data with 19 previously reported variants. Cross-referencing with clinical information revealed that 86% of people with GIGI-F1 mutations have autism. A similar proportion have communication difficulties and social phobia, alongside sleep problems and delayed speech. Zebrafish embryos edited via CRISPR to lack GIGI-F1 develop more slowly than their wild-type counterparts, Zhang and his colleagues found. As adults, the fish are more anxious and less willing to socialize. Zebrafish are typically highly sociable. They rarely swim alone, preferring to travel in clusters called shoals. When placed in a 3-chambered enclosure with a fish at one end and an empty chamber at the other, wild-type fish will opt for company. But fish lacking GIGI-F1 prefer solitude and appear anxious, frequently darting around the tank. What's more, they form looser shoals, maintaining more distance between themselves and others. Mice engineered to express just one copy of the GIGI-F1 gene also display social deficits. When the researchers tested for social novelty, a mouse's inclination to investigate a stranger over those it already knows, the mutant mice stayed near the familiar mouse. GIGI-F1 deletion also triggered repetitive behaviors, one of the core traits of autism. The study plays to the strengths of each model, says Julia Dahlman, associate professor of biology at the University of Miami in Florida, who was not involved in the study. For example, delayed embryogenesis is easier to spot in zebrafish, which develop outside their mother's body, than in mice. These different models can provide complementary windows of insight into gene function, she says. But the fish could be tweaked to better reflect the genetics of autism, says Dahlman. The study used zebrafish lacking both copies of GIGI-F1, but people with the condition have one mutated copy and one functional copy. Disrupting both gene copies is a good way to understand the biological importance of GIGI-F1, but does not model the human condition, Veenstra van der Wiel agrees. Mice missing a copy of the gene in only their excitatory neurons still showed repetitive behaviors and impairments in social novelty. Yet deleting a copy of GIGI-F1 in only inhibitory neurons triggered a different set of traits, including heightened anxiety and poorer cognition, suggesting that GIGI-F1 plays distinct roles in different cell types. The findings were published 14th of March in Biological Psychiatry. Exactly how GIGI-F1 variants cause changes in social behavior remains an open question. Right now, we only know a small piece of the whole picture. We still don't know the exact molecular mechanisms by which these mutations cause behavioral defects, Zhang says. Initial findings hint at changes in neuronal communication. By screening for transcripts and proteins that bind to GIGI-F1, the researchers identified hundreds of downstream targets, including several involved in synaptic transmission. They are now harnessing electrophysiology to see how GIGI-F1 deletion in mice influences neuronal chatter. Because GIGI-F1's function appears to be conserved among animal models, It would be interesting to see whether similar effects are seen in organoids and stem cells, says Katja Igreja, a researcher at the Max Planck Institute for Biology in Tübingen, Germany, who was not involved in the study. If so, scientists may be able to pinpoint GIGIF1's molecular mechanisms and identify potential therapeutic interventions that alleviate GIGIF1 deficiency, she adds.